Welcome to the deep dive. We take complex stuff and, well, we try to make it really clear.
That's the goal.
Today, we're jumping into something absolutely fundamental. How your nervous system, acting like this incredibly fast conductor, manages your circulation, especially blood pressure control.
Yeah, it's a fascinating system. And we've got a serious stack of sources here, textbooks, papers, all sorts. We're going deep into the anatomy, the mechanisms, the reflexes.
Everything that makes it tick.
Exactly. And the mission for this deep dive isn't just a quick overview. We want to pull out every key detail from these sources so you can really understand this at a professional level, truly master it.
Right, like getting the inside track. Okay, let's get into these sources and start unpacking how this all works.
Okay, so big picture first. Remember we talked about local tissue control before? Chapter 17 stuff? That's how blood flow adjusts within a specific organ based on its immediate metabolic needs.
Got it. Like local traffic control within a neighborhood.
Precisely.
Yay.
But the nervous system, that's operating on a much grander scale.
Yeah.
It handles the global functions.
Meaning?
Well, it's about things like rapidly redistributing blood flow around the whole body, changing how hard the heart pumps pretty dramatically, and, this is crucial, providing really fast control over your systemic arterial pressure. That rapid control is just vital.
And the main player doing this is the autonomic nervous system.
Almost entirely, yes. You've got the sympathetic and the parasympathetic branches. We'll see. The parasympathetic is absolutely essential for the heart.
Right.
But the sympathetic nervous system is really the main driver for controlling the blood vessels, the vasculature, pretty much everywhere else.
Okay, let's start there then. The sympathetic system. Where do these signals actually come from?
So these sympathetic nerve fibers, the ones controlling blood vessels, the vasomotor fibers, they originate in your spinal cord, specifically from all the thoracic segments and the first, maybe one or two lumbar segments.
From the spinal cord?
They pass into what are called the sympathetic chains. These are bundles of nerves running down either side of your spine, your vertebral column.
Okay. And how do they get from those chains out to the blood vessels?
Couple main routes. First, you have specific sympathetic nerves that branch off and go directly to innervate the internal organs, the viscera, gut, kidneys, and also directly to the heart itself.
Yeah.
The second route is that the fibers almost immediately join up with the regular peripheral spinal nerves, the ones going out to your arms and legs. And they travel with those nerves to reach the blood vessels in the periphery.
And our sources emphasize that the level of control, the innervation, isn't the same everywhere, right?
That's a really key detail. Most blood vessels get some sympathetic input, except for the capillaries, basically. But the density of these nerve endings varies a lot.
Where is it densest?
The small arteries, and especially the arterials, they have really dense innervation. And this is fundamental. Stimulating these nerves causes those small vessels to constrict powerfully. That's the primary way the nervous system increases resistance to blood flow.
So the arterioles are like the main control gates for blood flow and pressure under nervous control.
Exactly. Think of them as the taps. But it's not just them. The larger vessels, particularly the veins, are also significantly innervated.
And stimulating those.
Causes them to constrict too. Now, veins are quite floppy, so when they constrict, it reduces their volume, it effectively squeezes the blood stored in them.
Pushing it back towards the heart.
Precisely. Pushes it centrally. And that's incredibly important for regulating how much blood the heart has available to pump what we call venous return, which affects cardiac output. The sources also mention that even those tiny vessels right before the capillaries, the precapillary sphincters and metarterials, get some innervation in certain tissues, though usually less dense than the arterioles and veins.
So the nervous system can actively manage the blood reservoirs too. What about its direct effect on the heart?
Oh, it's huge. Sympathetic fibers go directly to the heart muscle, the conduction system, everything. Stimulation ramps up heart activity significantly.
How so?
Increases heart rate, sometimes dramatically, and it also enhances the actual force of contraction, makes the heart beat stronger and pump more blood with each beat. It's like hitting the accelerator on the heart.
Okay. Sympathetic. Constricts vessels, especially arterioles and veins, speeds up and strengthens the heart. Got it. Now the parasympathetic system. Sources say minor role in vessels, but crucial for the heart.
That's the main takeaway. For controlling blood vessels in most parts of the body, the parasympathetic system doesn't do much. Its really critical role in circulation is controlling heart rate.
And it does that via the vagus nerves.
Yes, primarily through the vagus nerves, those big nerves running from the brainstem right down to the heart.
And the effect is basically the opposite of the sympathetic.
Generally, yes. Parasympathetic stimulation via the vagus causes a marked decrease in heart rate. It also has a slight effect, decreasing the contractility, the force of the heart muscle, but the main effect is slowing the rate.
So sympathetic is the gas pedal for heart and vessels. Parasympathetic is the brake, mostly just for heart rate. Where's the central command post for all this?
That happens in a critical region in the brainstem called the vasomotor center. It's located on both sides, bilaterally, mainly in what's called the reticular substance of the medulla oblongata, and it extends down into the lower part of the pons as well.
And this center coordinates both branches, sympathetic and parasympathetic, for the circulation.
Exactly. It's the hub. It transmits those parasympathetic impulses down the vagus nerves to the heart, and it sends sympathetic impulses down through the spinal cord and out via the sympathetic nerves to control pretty much all the arteries, arterioles, and veins in the body.
Our sources break this vasomotor center down into different functional zones.
Yeah, based on stimulation experiments and mapping, we can identify 3 key areas. First, there's a vasoconstrictor area. This is in the anterolateral parts, sort of the front and side of the upper medulla. Neurons here are excitatory. They fire signals that excite those preganglionic sympathetic neurons down on the spinal cord that cause vasoconstriction.
Okay, that's the origin of the constrict signal.
That's the main driver. Second, there's a vasodilator area located just below it in the anterolateral parts of the lower medulla. Now, interestingly, these neurons project upwards to the vasoconstrictor area.
So they don't directly cause dilation.
No. They act by inhibiting the activity of the vasoconstrictor area. So when the vasodilator area fires, it reduces the firing of the constrictor area, which leads to less sympathetic output and thus vasodilation. It's an indirect effect.
Putting the brakes on the constrictor area makes sense. And the third area?
The sensory area. This is located in a structure called the nucleus tractus solitarius, which is in the posterolateral part, back inside, of the medulla and lower pons. This area is absolutely vital because it's the main receiving point for sensory information coming from the circulatory system itself.
Like signals from those pressure sensors we'll talk about.
Exactly. Signals from baroreceptors and chemoreceptors travel via the vagus and glossopharyngeal nerves and terminate right here in the nucleus tractus solitarius. The output signals from the sensory area then influence the activity of both the vasoconstrictor and vasodilator areas. This provides the anatomical basis for all those crucial circulatory reflexes, like the baroreceptor reflex.
So it's like the central processing unit gets input, analyzes it, tells the output areas what to do.
Now, this concept of sympathetic vasoconstrictor tone, what's that about? All right, this is really fundamental. Under normal resting conditions, the vasoconstrictor area isn't silent. It continuously transmits signals down to the sympathetic vasoconstrictor nerve fibers throughout the body.
Yeah.
It's not a massive burst, just a steady, slow stream of impulses, maybe half an impulse to 2 impulses per second.
It's like a constant background hum of activity.
Exactly. And this continuous low-level firing maintains a state of partial constriction in the blood vessels all the time. This baseline partial constriction is what we call vasomotor tone.
And it's important.
Hugely important. The sources highlight a classic experiment. If you use spinal anesthesia to block all sympathetic signals coming out of the spinal cord, you eliminate this tone completely. What happens? Arterial pressure plummets. It might drop from a normal 100 millimeter Hg down to maybe 50 millimeter Hg.
Wow.
It just shows how much of our normal resting blood pressure relies on this constant sympathetic tone. And if you then inject norepinephrine, the neurotransmitter, you can artificially restore that tone and bring the pressure back up.
That really illustrates its role. And the vasomotor center coordinates heart control simultaneously with this vascular tone.
Yes, it's a coordinated output. The lateral parts of the vasomotor center tend to send excitatory signals via sympathetic nerves to the heart, increasing heart rate and contractility. This often happens at the same time as increased vasoconstriction. Conversely, the medial portions of the center send signals to the vagal nuclei nearby, which then transmit parasympathetic impulses via the vagus nerves to the heart, which decreases heart rate and contractility.
So generally, constriction goes up, heart activity goes up, constriction goes down, heart activity goes down.
That's the general pattern, yes. The vasomotor center manages both the resistance vessels and the pump in a coordinated way.
And this control isn't just happening in the brainstem. Higher brain centers can influence the vasomotor center, too.
Oh, absolutely. Large numbers of neurons throughout the reticular substance higher up in the pons, the mesencephalon or midbrain, and the diencephalon, which includes the thalamus and hypothalamus, can either excite or inhibit the vasomotor center down in the medulla.
Any general rules?
Generally speaking, areas located more laterally and superiorly tend to be excitatory, while those more medially and inferiorly tend to be inhibitory.
And the hypothalamus. Sources say it's particularly powerful.
It really is. The hypothalamus can exert very strong excitatory or inhibitory effects on the vasomotor center. For example, stimulating the posterolateral parts usually causes strong excitation, leading to pressure increases. Stimulating parts of the anterior hypothalamus can cause either mild excitation or inhibition, depending on the exact spot.
What about the cerebral cortex, our thoughts and feelings?
Yes, even the cortex gets involved. Many parts can excite or inhibit the vasomotor center. The motor cortex, for instance, when you plan or initiate movement, tends to excite the vasomotor center, probably via pathways through the hypothalamus, and even more intriguingly, various areas deep in the base of the brain.
Like where?
Areas like the anterior temporal lobe, the orbital frontal cortex, the anterior cingulate gyrus, the amygdala, which is heavily involved in emotion, the septum, the hippocampus. Stimulating these areas can cause profound cardiovascular effects, either excitation or inhibition, depending on the specific spot and the stimulus.
So our emotional state, our planning, our memories even, can directly tweak our blood pressure and heart rate via these pathways.
It's a powerful connection, isn't it? Really
Right.
shows how integrated mind and body are, right down to circulatory control.
Okay, let's switch gears slightly. The sources mention a sympathetic vasodilator system. That sounds counterintuitive.
It does, doesn't it? Given the main job is constriction. But yes, sympathetic nerves going specifically to skeletal muscles actually carry some vasodilator fibers alongside the much more numerous constrictor fibers.
How does that work? What causes the dilation?
Well, in primates, including us, the thinking is it's mainly caused by circulating epinephrine. Remember, sympathetic stimulation triggers the adrenal glands to release epinephrine, adrenaline, into the blood. This circulating epinephrine can then stimulate specific receptors on the blood vessels in skeletal muscle. Beta-adrenergic receptors and activation of these receptors causes the muscle to relax, leading to vasodilation.
So it's the hormone acting, not directly the nerve-ending transmitter in this case.
That's the dominant theory for primates. In some other animals, there's evidence for acetylcholine being released by these sympathetic nerves, which might cause vasodilation, perhaps via nitric oxide. But for us, epinephrine seems key.
And which brain area controls this vasodilator outflow?
The main area identified seems to be the anterior hypothalamus.
Okay, so how important is this system in humans, really? The sources seemed a bit hesitant.
Yeah, the consensus seems to be it doesn't play a major role in the overall moment-to-moment control of circulation. If you block these sympathetic nerves to muscles, it doesn't massively disrupt blood flow regulation under many normal conditions.
But maybe it has a specific role.
Possibly. Some research suggests it might be important right at the start of exercise. It could contribute to an anticipatory vasodilation in the muscles, kind of preparing them for the increased blood flow they're about to need. This could be driven by that initial surge of epinephrine hitting the beta receptors, or maybe some acetylcholine effect.
Like opening the gates a little just before the rush?
Okay.
Now, another interesting situation involving vasodilation is fainting due to emotion vasovagal syncope.
Right, that's a really striking phenomena during intense emotional distress. Fear, shock, sometimes even seeing blood, you can get this maladaptive response. The muscle vasodilator system we just talked about becomes activated.
So vessels in the muscles relax.
Yes, and at the same time, the vagal cardioinhibitory center in the brainstem sends out incredibly strong signals via the vagus nerves to the heart.
What does that do to the heart?
Drastically slows it down. So now you have widespread vasodilation, mainly in the muscles, which drops your peripheral resistance, and a very slow heart rate, which drops your cardiac output.
Both together would plummet blood pressure.
Exactly. Arterial pressure falls rapidly and dramatically. Blood flow to the brain becomes insufficient, and you lose consciousness, you faint. The pathway likely involved those higher brain centers, the cortex processing the emotional event, somehow triggering the anterior hypothalamus, the vasodilatory center, and also strongly activating the vagal centers in the medulla, leading to both the vasodilation via sympathetic nerves and the cardioinhibition via the vagus nerves.
Wow. A real system short circuit caused by emotion. Okay, let's return to the core job of the nervous system.
Yeah.
Rapid control of arterial pressure itself.
Right. This is arguably its most vital, rapid function. The nervous system has this incredible capacity to cause very fast increases in arterial pressure when needed.
How does it achieve that?
It does it by activating the entire vasoconstrictor and cardioaccelerator parts of the sympathetic system together as a coordinated unit. Full throttle sympathetic drive.
And turning off the brakes.
And at the same time, importantly, it inhibits the parasympathetic vagal signals that would normally slow the heart down. So you remove the brake while hitting the gas. This combination triggers three major things simultaneously, all working to push pressure up fast.
Okay, what are the three?
First, you get strong constriction of most of the arterioles throughout the body. This massively increases total peripheral resistance, like clamping down on the outflow pipes. That directly raises pressure.
Makes sense. Number two.
Second, you get strong constriction of the veins and other large capacitance vessels. This is key. It squeezes blood out of these peripheral reservoirs, pushing it back towards the heart. This increases the volume of blood filling the heart chambers.
Increased filling means?
The heart muscle gets stretched more. And due to the Frank-Starling mechanism, it responds by contracting more forcefully, pumping out more blood. So, venous constriction boosts cardiac output.
Okay, increased resistance, increased return to the heart. What's third?
Third is the direct sympathetic stimulation of the heart itself. This further enhances its pumping. Heart rate increases significantly, maybe up to 3 times normal in strong stimulation. And the direct stimulation increases the heart muscle's contractile force, potentially doubling how strongly it can pump.
So higher resistance, more blood returning, and a faster, stronger pump. That's a powerful combination.
Extremely powerful. And the speed is just remarkable. The response starts within seconds. Pressure can literally double in just 5 to 10 seconds. And the flip side is also fast. If you suddenly inhibit sympathetic outflow, pressure can drop by half in maybe 10 to 40 seconds. It's easily the most rapid mechanism we have for pressure regulation.
Which is perfect for sudden demands like exercise or stress.
Absolutely. Take heavy exercise. Your muscles need way more blood flow. Some of that comes from local factors causing vasodilation in the muscles.
Right, matching flow to metabolic need locally.
But you also get a big boost because the nervous system simultaneously elevates the overall arterial pressure system-wide.
How much does pressure go up during exercise?
Sources say typically 30% to 40% during heavy exercise. That higher pressure driving the blood, combined with the local dilation in the muscles, helps achieve that massive increase in muscle blood flow, sometimes nearly doubling it just from the pressure effect alone.
And this pressure rise is mainly driven by the nervous system.
Primarily, yes. When your brain's motor areas activate to make you move, they also send signals, partly via the reticular activating system, that excite the vasoconstrictor and cardioaccelerator areas in the vasomotor center. It's like the brain anticipates the need and primes the circulatory system, ensuring pressure rises quickly to meet the demand.
And similar things happen in other stressful situations. Fright, for example.
Yes, the so-called alarm reaction. Extreme fright can cause pressure to skyrocket, maybe 75 to 100 millimeter Hg, in just seconds. This provides that immediate high pressure needed to perfuse the muscles for a potential fight or flight response.
Okay, so that covers the rapid responses to demands. But the nervous system also works constantly in the background to keep pressure stable, right? Through reflexes.
Exactly. Beyond these big acute responses, there are multiple subconscious negative feedback reflexes working continuously to keep arterial pressure hovering around its normal set point. The most famous and arguably most important of these, at least for short-term control, is the baroreceptor arterial pressure control system, the baroreflex.
Triggered by those stretch receptors.
That's the one. Baroreceptors are specialized nerve endings sensitive to stretch, located in the walls of certain large systemic arteries. When your arterial pressure goes up, it stretches the artery walls more, and this stimulates the baroreceptors.
And they send signals. Where?
They send signals back to the central nervous system, specifically to that sensory area, the nucleus tractus solitarius, and the dula. In response, the CNS sends signals out via the autonomic nervous system to bring the pressure back down towards normal. It's a classic negative feedback loop.
Where exactly are these key baroreceptors found?
They're described as spray-type nerve endings. While they exist in many large arteries, the really critical concentrations are in 2 spots. First, in the wall of the internal carotid artery, just above where it branches off the common carotid, that area is called the carotid sinus, and second, in the wall of the aortic arch.
Good locations monitoring pressure going to the brain via the carotids and pressure going to the rest of the body via the aorta. How do the signals get from there to the brainstem?
Signals from the carotid sinus baroreceptors travel up through small nerves called Herring's nerves, which then join the glossopharyngeal nerves, that's cranial nerve 9, and they travel into the medulla, ending in the nucleus tractus solitarius.
Okay, and the aortic ones?
Signals from the aortic arch baroreceptors travel up via the vagus nerve's cranial nerve 10, also ending in the very same nucleus tractus solitarius. So all that pressure information converges in that one sensory hub.
How sensitive are they? Is it just on or off?
No, it's a graded response, and this is really key to understand. Let's take the carotid ones. They don't fire much at all below pressures of, say, 50 or 60 millimeter Hg. But as pressure rises above that, their firing rate increases progressively. They become increasingly active up to about 180 millimeter Hg, where they max out.
So they cover a wide range.
They do. The aortic baroreceptors are similar, but generally operate at pressures about 30 millimeter Hg higher. But the crucial point is where they are most sensitive. Right around the normal mean arterial pressure, around 100 millimeter Hg, even a very small change in pressure causes a large change in their firing rate. That makes the reflex incredibly effective at correcting deviations right around the normal set point.
Tuned perfectly for maintaining normal pressure, and they respond quickly.
Extremely quickly. Within a fraction of a second, you can actually see their firing rate increase during systole when pressure peaks and decrease during diastole when it falls with each heartbeat. And another key point from the sources, they respond more strongly to a rapidly changing pressure than to a steady pressure.
What do you mean?
Well, if pressure is quickly rising towards 150 millimeter Hg, the baroreceptors might fire twice as fast as they do when the pressure is just sitting steady at 150 millimeter Hg. They sense the rate of change as well as the absolute level.
Like predicting where it's going. Okay, so the signals arrive at the nucleus tractus solitarius. What's the downstream effect? What does the reflex do?
When the baroreceptors are stimulated by high pressure, the signals coming out of the nucleus tractus solitarius do two main things. They inhibit the vasoconstrictor center in the medulla, and they excite the vagal parasympathetic center.
Okay, so high pressure leads to less sympathetic constriction and more parasympathetic slowing of the heart.
Exactly. The net effects are 1. Vasodilation of arterioles and veins throughout the body, which decreases total peripheral resistance, and 2. Decreased heart rate and decreased strength of heart contraction, which decreases cardiac output. Both of these actions, lower resistance and lower output, work together to bring the elevated arterial pressure back down towards normal.
And if pressure falls too low?
The opposite happens. Low pressure means less baroreceptor firing. This leads to less inhibition of the vasoconstrictor center, so it becomes more active. It also leads to less excitation of the vagal center, so parasympathetic output decreases. The result: increased sympathetic output causing vasoconstriction, increased venous return, increased heart rate, and increased heart contractility. All push the pressure back up.
That classic experiment where they clamp the carotids really shows this powerfully.
It does. If you temporarily clamp the common carotid arteries below the sinus, you drastically reduce the pressure inside the carotid sinus. The baroreceptors there sense this low pressure and decrease their firing rate dramatically.
Which tells the brain pressure is low.
Right. So the brain removes the inhibition from the vasomotor center, which becomes highly active. Systemic arterial pressure, measured elsewhere, shoots up and stays high as long as the clamps are on. Then, when you release the clamps, pressure in the sinus goes back to normal, the baroreceptors fire strongly again, and systemic pressure quickly drops, often even overshooting slightly below normal for a moment before stabilizing. It perfectly demonstrates the negative feedback loop.
And this reflex is working all the time, like when we just stand up.
Absolutely critical for that. When you stand up, gravity pulls blood down into your legs and abdomen. This immediately lowers pressure in your upper body and head. Without the baroreflex, you'd likely get dizzy or faint every time you stood up.
So what happens?
The falling pressure in the carotid sinus and aortic arch is instantly detected. Baroreceptor firing drops. This triggers an immediate, powerful reflex sympathetic discharge throughout the body. Widespread vasoconstriction, increased heart rate, it all counteracts the effect of gravity, minimizing the pressure drop in your head and keeping you conscious.
It's constantly buffering these changes. That's why they call it the pressure buffer system.
Exactly. And the nerves carrying the signals are the buffer nerves. And you really see how important this buffering is if the nerves are cut. The sources describe experiments in animals where, after denervating the baroreceptors, arterial pressure becomes incredibly labile, incredibly variable.
How variable?
Huge swings. Pressure might range from 50 millimeter Hg up to 160, or even higher throughout the day, just fluctuating wildly with normal activities like sleeping, eating, excitement, posture changes. Compared to an animal with intact baroreceptors where pressure stays in a much narrower band, say 85 to 115 millimeter Hg, the reflex essentially cuts down the minute-to-minute pressure variation by about 2/3. It smooths everything out.
Okay, clearly crucial for short-term, moment-to-moment stability. But what about long-term pressure regulation? The sources seem to suggest their role there is a bit more complicated.
Yes, that's been an area of debate. A key reason is that baroreceptors tend to reset or adapt over time. If your blood pressure becomes chronically elevated, say you develop hypertension and your pressure stays around 160 millimeter Hg,
Hmm.
initially the baroreceptors fire much faster because of the high pressure, but over the next day or two, even though the pressure stays high, their firing rate gradually drifts back down towards the normal rate for 100 millimeter Hg. They adapt to the new higher baseline. The same happens in reverse if pressure is chronically low.
So they start treating the new pressure as normal.
In a sense, yes. And this resetting phenomenon led people to think they might not be very effective in correcting long-term pressure problems like chronic hypertension.
Mm,
If they just adapt, how can they drive the pressure back down persistently?
makes sense. But is that the full story now?
It seems not entirely. More recent understanding, reflected in our sources, suggests they do contribute to long-term regulation, even with resetting. Firstly, the resetting might not be 100% complete. Secondly, and perhaps more importantly, they have a sustained influence on sympathetic nerve activity going specifically to the kidneys.
Ah, the kidney connection again.
Right. Even with resetting, chronically elevated pressure sensed by the baroreceptors seems to lead to a sustained decrease in sympathetic signals going to the kidneys.
And less sympathetic drive to the kidneys
Means the kidneys are signaled to excrete more sodium and water. This leads to a gradual reduction in blood volume over days and weeks, which is a powerful mechanism for lowering arterial pressure long-term. So the baroreceptors likely play a crucial long-term role by modulating kidney function, interacting with the body's main long-term pressure control system, the renal body fluid system.
So it's a partnership for long-term control.
It seems so. And studies using chronic electrical stimulation of the baroreceptor nerves, which can cause sustained reductions in blood pressure, also support the idea that they retain significant long-term influence. It suggests maybe a lot of the resetting we see in hypertension happens at the level of the receptors themselves adapting, rather than the central brain circuits becoming completely insensitive.
Fascinating how that understanding has evolved. Okay, alongside baroreceptors, we also have chemoreceptors involved in pressure control.
Yes, another reflex system closely associated geographically with the baroreceptors, but responding to chemical changes, not stretch.
What chemicals?
They're primarily sensitive to low oxygen levels, hypoxia, high carbon dioxide levels, hypercapnia, and increased acidity, meaning high hydrogen ion concentration.
And where are these located?
In tiny structures just a couple of millimeters across called chemoreceptor organs. There are two carotid bodies located near the carotid sinus at the bifurcation of the common carotid arteries. And there are usually 1 to 3 aortic bodies located near the aortic arch, close to the aortic baroreceptors.
And their signals travel back to the brainstem, too.
Yes, they excite nerve fibers that travel along with the baroreceptor signals through Herring's nerves from the carotid bodies, and the vagus nerves from the aortic bodies ending up, again, in the nucleus tractus solitarius in the medulla.
When do these chemoreceptors actually kick in for blood pressure control?
They have an incredibly high blood flow relative to their size. This makes them very sensitive to any reduction in that flow. They typically become significantly stimulated when arterial pressure falls below a certain level, usually around 80 millimeter Hg.
Why then?
Because when pressure and flow drop that low, oxygen delivery to the chemoreceptors decreases, and maybe more importantly, waste products like CO2 and acid aren't washed away effectively. This chemical change, low O2, high CO2, high acid, activates the receptors.
And what's the reflex response?
The signals generated by the stimulated chemoreceptors strongly excite the vasomotor center. This increases sympathetic output, causing vasoconstriction and increased heart activity, which elevates arterial pressure back up towards normal.
So they help boost pressure when it gets too low.
Exactly. But a key point here is that they are not a powerful controller of pressure above 80 millimeter Hg. Their main significance for blood pressure regulation is really in that lower pressure range below 80. Down there, they become a really important, powerful reflex to prevent pressure from falling further into dangerous territory.
Kind of like an emergency backup system for low pressure situations.
That's a great way to think about it. Their primary job is actually respiratory control, responding to blood gas levels to adjust breathing, but they definitely provide this crucial cardiovascular support when pressure drops significantly. And their activation might also contribute to higher pressure in conditions like severe obesity or sleep apnea where blood oxygen can drop.
Okay, so we have high pressure sensors, baroreceptors, and low pressure chemical sensors, chemoreceptors, in the arteries. What about sensors in the low pressure parts of the circulation, like the atria and pulmonary arteries?
Yes, absolutely. There are stretch receptors located in the walls of the atria of the heart and also in the pulmonary arteries. These are often collectively called low-pressure receptors. They function similarly to the arterial baroreceptors, sensing stretch, but they're situated in the low-pressure side of the system.
What triggers them?
They primarily respond to increases in pressure in these areas, which usually happens when your total blood volume increases. Think about getting an IV fluid infusion. That extra volume stretches the atria and pulmonary vessels.
And what's their role?
They play a really important role in minimizing the changes in arterial pressure that result from changes in blood volume. The sources give an example. Infuse blood into an animal and its arterial pressure will rise. But that pressure rise is much, much smaller if the low-pressure receptors and the arterial baroreceptors are working properly compared to an animal where these receptors have been denervated.
So they act as another buffer system, specifically buffering the effects of volume changes on arterial pressure.
Precisely. They initiate reflexes that run parallel to the arterial baroreceptor reflexes, making the whole system more effective at controlling pressure, especially when blood volume fluctuates.
And these atrial reflexes are particularly involved in signaling the kidneys to deal with excess volume.
Yes, this is a major function, often called the volume reflex. When the atria gets stretched by increased blood volume, these low-pressure receptors fire. Their signals lead to several things aimed at getting rid of the excess fluid,
Such as
reflex decreases in sympathetic nerve activity going to the kidneys. This leads to less reabsorption of sodium and water by the kidney tubules. They also cause dilation of the afferent arterioles, the small arteries bringing blood into the filtering units of the kidneys, the glomeruli.
How does dilating those arterioles help?
It increases blood flow into the glomeruli and raises the pressure inside them, which drives more fluid filtration out of the blood and into the tubules, the first step in making urine.
So less reabsorption, more filtration. Any other effects?
Yes, signals from the stretched atria also go up to the hypothalamus in the brain and decrease the secretion of antidiuretic hormone, or ADH. Less ADH means the kidneys reabsorb less water, so you have increased fluid filtering out and decreased water being pulled back in. The net result is significantly increased urine output, losing the excess fluid.
And isn't there a hormone released by the atria themselves, AMP?
Exactly. Atrial natriuretic peptide, that's another key part. The physical stretch of the atrial muscle cells also causes them to release AMP into the bloodstream. AMP acts directly on the kidneys to further promote the excretion of both sodium and water.
So it's a multi-pronged attack on excess volume driven by atrial stretch?
It is. All these mechanisms work together to bring blood volume back towards normal. And by controlling volume, they indirectly help control pressure too, since excess volume tends to increase cardiac output and therefore pressure. It's crucial for long-term fluid balance and pressure regulation.
Okay. Now, increased atrial pressure can also directly affect heart rate through something called the Bainbridge reflex.
Yes, another reflex originating from atrial stretch. Increased atrial pressure or volume can trigger an increase in heart rate, sometimes by as much as 75%. This effect seems most prominent when the initial heart rate is relatively slow.
Wait, so increased volume stretches the atria, which increases heart rate via Bainbridge, but increased volume also raises arterial pressure, which should decrease heart rate via the baroreflex. Seems contradictory.
It can be. There's a potential interplay there. The overall change in heart rate when blood volume increases depends on the balance between the Bainbridge reflex pushing the rate up and the arterial baroreflex trying to pull it down. However, the sources suggest that especially with significant volume increases, the Bainbridge reflex often predominates, leading to a net increase in heart rate.
How does the Bainbridge reflex actually work?
A small part of the rate increase, maybe 15%, might come from the increased volume directly stretching the heart's natural pacemaker, the sinus node. But the main effect, maybe 40–60% increase, is a true reflex. Stretch receptors in the atria send signals via the vagus nerves up to the medulla. Then, efferent signals come back down via both vagal, less inhibition, and sympathetic nerves to increase heart rate and also the strength of contraction.
What's the point of speeding up the heart when volume increases?
The idea is it helps prevent blood from damming up or backing up in the veins, atria, and pulmonary circulation. By pumping faster and stronger, the heart can more effectively move that increased volume forward through the system.
Okay, we've covered reflexes starting from arteries, atria, pulmonary vessels. What if the problem isn't out there but right in the control center? What if blood flow to the brainstem itself fails?
That triggers the most powerful sympathetic response of all, the CNS ischemic response. This isn't initiated by peripheral receptors. It happens when blood flow to the vasomotor center in the lower brainstem becomes so severely reduced that the neurons there start to suffer from lack of oxygen and nutrients, they become ischemic.
Because the control center itself is being starved.
Exactly. And when the vasoconstrictor and cardioaccelerator neurons in the vasomotor center become ischemic, they get directly and powerfully excited by this pathological state.
Not a reflex arc, but a direct, desperate activation.
Precisely. And the result is a massive, system-wide sympathetic discharge, causing arterial pressure to shoot up to extremely high levels, sometimes as high as the heart can physically pump, maybe 250 millimeters HG or even more for short periods.
What causes this intense excitation during ischemia?
The leading theory is the buildup of carbon dioxide. When blood flow is severely reduced, CO2 produced by the brainstem neurons can't be washed away. CO2 concentration rises dramatically locally, and CO2 is an incredibly potent stimulator of the sympathetic vasomotor neurons. Other waste products like lactic acid might also contribute.
The CNS ischemic response sounds incredibly drastic. Is it part of normal regulation?
Absolutely not. It's purely an emergency life-saving mechanism. It doesn't kick in until arterial pressure falls to very low levels, typically below 60 millimeters Hg, and it becomes maximal at critically low pressures, maybe 15-20 millimeters Hg. Its sole purpose is to try and restore blood flow to the vital brainstem centers when perfusion is so low that it's threatening survival. It's often called the last-ditch stand response.
A final attempt to save the control centers, and the Cushing reaction is related to this.
Yes, the Cushing reaction is a specific type of CNS ischemic response. It's triggered not by low systemic blood pressure, but by increased pressure of the cerebrospinal fluid, CSF, within the skull. If CSF pressure rises high enough due to, say, a tumor or swelling, it can physically compress the arteries supplying the brain, cutting off blood flow.
Causing brain ischemia that way.
Right. This ischemia in the brainstem then triggers the CNS ischemic response. Systemic arterial pressure rises dramatically, crucially, rising above the level of the elevated CSF pressure.
Why is that important?
Because once arterial pressure exceeds CSF pressure, blood can again be forced into the cerebral arteries, restoring flow and relieving the ischemia. The system then finds a new equilibrium where arterial pressure stays just slightly higher than the CSF pressure. It's a protective mechanism to ensure the brain gets perfused even when intracranial pressure is dangerously high.
So the nervous system uses autonomic pathways, but skeletal nerves and muscles also play a role in pressure control.
They do, in specific situations. Our sources highlight two important examples where skeletal muscle activity significantly impacts circulation and pressure.
One is the abdominal compression reflex.
That's right. This reflex can be triggered by signals from the baroreceptors or chemoreceptors. But instead of just autonomic output, nerve signals are also sent via skeletal motor nerves to the abdominal muscles.
Making the abs contract.
Yes, causing strong contraction of the abdominal wall muscles. This squeezes the large veins and other blood reservoirs within the abdomen, effectively pushing blood out of the abdomen and towards the heart.
Like squeezing a sponge.
Exactly. This increases venous return, boosts cardiac output, and helps raise arterial pressure. It has a similar effect to sympathetic constriction of veins, but uses skeletal muscle power. The sources note this might be more important than we used to think, as people with paralysis of these muscles often have more trouble with low blood pressure.
That makes sense. What's a second example involving skeletal muscle?
That's simply the effect of muscle contraction during exercise. Whenever you use your muscles, even just tensing them, they compress the blood vessels running through and around them.
A mechanical squeezing effect.
Yes. This happens throughout the body during exercise. This widespread muscle contraction literally milks blood out of the peripheral veins and towards the heart and lungs, significantly boosting venous return. This mechanical pumping action by the muscles is absolutely essential to achieve the huge increases in cardiac output, maybe 5 to 7 times resting levels needed during heavy exercise. And that increased output is a major factor contributing to the rise in arterial pressure we see during exercise.
So voluntary muscle action provides a crucial mechanical assist to circulation during activity.
Okay, we've covered these major control mechanisms. What about those smaller rhythmic fluctuations you sometimes see on a blood pressure trace? Respiratory waves.
Right. Arterial pressure doesn't usually stay perfectly flat. It tends to rise and fall slightly with each breath. These are respiratory waves, typically around 4 to 6 millimeters Hg in magnitude.
What causes them? It's likely a combination of factors. One is spillover, signals from the brain's respiratory center, influencing the nearby vasomotor center rhythmically. Another is purely mechanical. When you inhale, the pressure inside your chest becomes more negative. This causes the large veins and pulmonary vessels in the chest to expand slightly, which can momentarily reduce the amount of blood returning to the left heart, causing a brief dip in cardiac output and pressure.
And maybe stretch receptors are involved too?
Possibly. The pressure changes in the chest vessels during breathing could also rhythmically stimulate atrial or vascular stretch receptors, contributing to the fluctuations via reflex pathways. The net result is usually a slight rise in pressure during early exhalation and a fall during the rest of the cycle. With deep breathing, these waves can become much larger, maybe 20 millimeter Hg.
And then there are even slower, larger waves, vasomotor waves or Mayer waves?
Yes, these are bigger swings in pressure, maybe 10 to 40 millimeter Hg, and they happen much more slowly, with a cycle length of perhaps 7 to 10 seconds in humans. Figure 1811 shows examples.
What drives these slower oscillations?
These seem to be caused by the oscillation, or hunting, of the nervous pressure control reflexes themselves, primarily the baroreceptor and chemoreceptor reflexes.
How does a reflex oscillate?
Think about the baroreflex negative feedback loop. High pressure triggers a response to lower pressure, but there's a time delay between sensing the high pressure and the effect kicking in. Because of this delay, the correction might overshoot, causing pressure to fall too low.
Okay.
Now, the low pressure reduces baroreceptor firing, triggering a response to raised pressure. Again, due to delay, this correction might overshoot, setting pressure too high, and the cycle repeats. The system oscillates around the set point because the feedback gain is high and there's a delay. The chemoreceptor reflex can oscillate similarly, especially in the lower pressure ranges where it's more active.
So the control system itself can create these waves because of inherent delays.
Precisely. The sources even show that the powerful CNS ischemic response can oscillate, especially under conditions like high CSF pressure. The ischemic response drives pressure up, relieving the ischemia. This turns off the response, pressure falls, causing ischemia again, which restarts the response. It cycles. The core principle is that any powerful feedback control system with a time delay can potentially oscillate if the feedback is strong enough. It's a classic control systems phenomenon seen in engineering too, but happening right here in our circulatory control.
Wow. We have covered an incredible amount of detail in this deep dive. Seriously, from the sympathetic nerve anatomy, the different receptors, the vasomotor center structure, all the way to these complex reflexes like baroreceptors, chemoreceptors, the volume reflex, and even that extreme CNS ischemic response.
That's a lot, isn't it? We've seen how the nervous system isn't just making tiny adjustments. It orchestrates these really rapid, powerful, system-wide changes in pressure and flow distribution. It handles everything from the simple act of standing up to meeting the demands of intense exercise to surviving life-threatening situations.
It's this incredibly dynamic interplay. Constant background buffering from the baroreceptors, readiness for rapid action via the sympathetic system, coordination with volume control via the kidneys and atrial reflexes. It's all happening simultaneously, mostly subconsciously. To really get a handle on this, you need to appreciate all those layers, the nerve pathways, the specific receptors where they send signals in the brainstem, and how those feedback loops operate with such speed.
Exactly. And thinking about all of it together, the sheer speed of these responses, measured in seconds, the power of something like the sympathetic surge or the CNS ischemic response, the constant fine-tuning required just to keep pressure stable. What aspect of this complex system really stands out to you now? Knowing all this, does it change how you think about, say, everyday stress, or even just the simple act of getting out of bed in the morning and what your body's doing automatically to manage it?
Definitely gives you a new appreciation for the constant background activity. Until next time, keep diving deep.